Chapter 7. The Endocrine System. This chapter focuses on the role of hormones in regulating various physiological processes and how the endocrine system maintains homeostasis through chemical signaling. Hormones are chemical messengers produced by endocrine glands that travel through the bloodstream to target tissues. They regulate functions such as growth, metabolism, reproduction, and mood. The classification of hormones are classified based on their chemical structure and how they interact with target cells. The main categories include peptide hormones, steroid hormones, and amine hormones, each with distinct mechanisms of action and duration of effects. Hormone secretion is regulated through complex feedback mechanisms to maintain balance within the body. Stimuli triggering hormone release include neural signals, hormonal signals from other glands, and changes in blood levels of certain ions or nutrients. Hormones can interact in several ways at target tissues. They may work synergistically to enhance effects, act antagonistically to oppose each other, or demonstrate permissiveness, where one hormone allows another to exert its full effect. Disorders of the endocrine system arise from hormone excess, deficiency, or abnormal receptor function. These pathologies can disrupt normal physiological processes, leading to conditions such as diabetes mellitus, hyperthyroidism, or adrenal insufficiency. Endocrinology is the scientific study of hormones, which are chemical messengers produced by endocrine glands that play a critical role in regulating the body's long-term, ongoing functions. These functions include metabolism, the regulation of the internal environment, reproduction, growth, and development. Hormones act in three basic ways to maintain homeostasis and coordinate physiological processes. First, they can augment the rates of enzymatic reactions, increasing or decreasing the speed of biochemical processes. Second, hormones regulate the transport of ions or molecules across cell membranes, controlling nutrient uptake, ion balance, and cellular communication. Third, they influence gene expression and protein synthesis, altering the production of proteins that are essential for cellular function and tissue development. By understanding how hormones operate, it becomes clear how the endocrine system plays a pivotal role in maintaining balance and supporting the body's vital functions. A hormone is a chemical signal that facilitates communication between different parts of the body. Hormones are secreted by a single cell or a group of cells and are released into the bloodstream. This process begins with the secretion of the chemical signal from a cell into the extracellular fluid, where it diffuses into the blood. Once in the bloodstream, hormones are transported to distant target tissues or organs where they exert their effects. Despite being present in very low concentrations, hormones are highly effective in initiating physiological responses. This allows them to regulate a variety of essential processes such as growth, metabolism, and homeostasis with remarkable precision. Hormones exert their effects by binding to specific receptors located on or within target cells. The cellular mechanism of action relies on this binding process, as it triggers biochemical responses that influence the cell's function. When a hormone binds to its receptor, it can activate or inhibit various cellular pathways, ultimately affecting gene expression, enzyme activity, or ion transport. To maintain homeostasis, hormone action must be terminated once the desired response is achieved. This termination is often measured by the hormone's half-life, which refers to the time it takes for half of the hormone's concentration in the blood to be eliminated. A shorter half-life indicates rapid hormone clearance, while a longer half-life suggests prolonged activity, ensuring that hormonal signals are appropriately regulated to prevent excessive or insufficient responses. Hormones are classified based on their chemical structure, with most being peptides or proteins. These hormones are synthesized from amino acids and are water-soluble, allowing them to be easily transported in the bloodstream. However, they have a relatively short half-life compared to steroid hormones, meaning they are quickly broken down and cleared from the body. The cellular mechanism of action for peptide hormones involves binding to receptors located on the surface of the target cell membrane. Since these hormones cannot pass through the lipid bilayer, they rely on membrane-bound receptors to initiate their effects. Upon binding, a signal transduction system is activated, leading to a cascade of intracellular events that ultimately produce the desired cellular response. This process allows for rapid and specific regulation of physiological functions such as metabolism, growth, and immune responses. Peptide hormones, which are water-soluble, cannot enter their target cells directly. Instead, they bind to specific membrane receptors located on the surface of target cells to initiate a signal transduction process. Upon binding to the receptor, the interaction activates a series of intracellular events. One common pathway involves the activation of G proteins, which in turn stimulate amplifier enzymes. These enzymes catalyze the formation of second messenger molecules within the cell. These second messengers, such as cyclic adenosine monophosphate, propagate the signal by activating protein kinases. Protein kinases phosphorylate target proteins, altering their function and leading to a cellular response. In some cases, peptide hormones bind to receptors with intrinsic enzyme activity, such as tyrosine kinase receptors. When activated, these receptors directly phosphorylate proteins without the need for second messengers, resulting in rapid cellular changes. This signal transduction system ensures that extracellular signals from peptide hormones are converted into appropriate intracellular actions, controlling various physiological processes like metabolism, gene expression, and ion channel regulation. Steroid hormones are synthesized from cholesterol and are produced in specific organs, primarily the adrenal cortex of the adrenal glands and the gonads. These hormones play crucial roles in regulating metabolism, immune response, salt and water balance, and reproductive functions. In the bloodstream, steroid hormones bind to carrier proteins to increase their solubility and prolong their half-life. For example, cortisol, a glucocorticoid hormone, has a half-life ranging between 69 to 90 minutes in circulation due to its association with binding proteins. The cellular mechanism of action of steroid hormones involves their ability to pass through the lipid bilayer of the cell membrane due to their lipophilic nature. Once inside the cell, steroid hormones bind to intracellular receptors located in the cytoplasm or nucleus. This hormone receptor complex then binds to specific regions of DNA, initiating or inhibiting the transcription of target genes. These genomic effects result in the synthesis of proteins that carry out various physiological functions, often leading to long-lasting cellular changes. Cholesterol serves as the parent compound for all steroid hormones. These hormones are primarily produced in the adrenal cortex and gonads, which include the ovaries and testes. Due to their lipophilic nature, steroid hormones are not stored within endocrine cells. Instead, they are synthesized on demand and diffuse out of the cell immediately after production. The synthesis pathway begins with cholesterol being converted into various intermediate compounds, eventually leading to the formation of different steroid hormones. Dehydroepiandrosterone, also known as DHEA, and androstenedione lead to the production of androgens like testosterone. Testosterone can be further converted into dihydrotestosterone or aromatized into estradiol. Progesterone is converted into corticosterone, which can either produce aldosterone, involved in sodium balance, or convert into cortisol, which plays a role in the stress response and metabolism. Androstenedione can also be converted into estrone and further into estradiol, both of which are essential for reproductive functions. Each step in the pathway is catalyzed by specific enzymes, such as 21-hydroxylase and aromatase, to ensure the production of the appropriate hormone depending on the body's needs. These hormones play crucial roles in metabolism, reproduction, immune response, and the regulation of electrolyte balance. Steroid hormones primarily act on intracellular receptors. Most hydrophobic steroid hormones circulate in the bloodstream bound to plasma protein carriers, with only the unbound hormones able to diffuse into target cells. Once inside, these hormones bind to receptors located in the cytoplasm or nucleus. Some steroid hormones also interact with membrane-bound receptors, triggering rapid cellular responses through second messenger systems. Upon binding to its receptor, the hormone receptor complex moves into the nucleus, where it attaches to specific DNA sequences. This interaction activates or represses the transcription of one or more genes. The newly transcribed messenger RNA exits the nucleus and enters the cytoplasm, where it directs the synthesis of new proteins through translation. These newly formed proteins carry out various functions essential for cellular processes and the body's physiological responses. Some hormones originate from single amino acids. Those derived from tryptophan include melatonin, which is secreted by the pineal gland and plays a crucial role in regulating sleep-wake cycles. Hormones derived from tyrosine can be categorized into two groups. The first group includes catecholamines such as epinephrine, norepinephrine, and dopamine. These hormones function similarly to peptide hormones and are involved in the body's stress response, blood pressure regulation, and mood control. The second group consists of thyroid hormones, which are formed from two tyrosine molecules. Unlike catecholamines, thyroid hormones act like steroid hormones by exerting genomic effects that regulate metabolism and growth by influencing gene expression. Amine hormones are primarily derived from the amino acid tyrosine. Tyrosine serves as the precursor for both catecholamines and thyroid hormones. Catecholamines are synthesized by modifying the side groups of tyrosine and include dopamine, norepinephrine, and epinephrine. These hormones are involved in regulating mood, stress responses, and cardiovascular functions. Thyroid hormones, formed from two tyrosine molecules and iodine atoms, include thyroxine, or tetraiodothyronine, and triiodothyronine. These hormones are crucial for regulating metabolism, growth, and development by influencing gene expression. Reflex pathways provide a method to understand how hormones are released. All reflex pathways share components: stimulus, sensor, input signal, integration, output signal, targets, and response. In simple endocrine reflexes, the endocrine cell itself acts as the sensor. An example is the secretion of parathyroid hormone, which regulates calcium levels. Many endocrine reflexes involve the nervous system. For instance, the parasympathetic nervous system can stimulate insulin secretion. Neurohormones are also involved in endocrine reflexes, with examples including antidiuretic hormone and oxytocin, both of which play roles in fluid balance and reproductive functions. The left diagram shows a simple endocrine reflex pathway for parathyroid hormone release. A decrease in plasma calcium concentration stimulates the parathyroid gland to secrete parathyroid hormone. The hormone acts on bones and kidneys to increase plasma calcium levels through bone resorption, kidney reabsorption of calcium, and increased intestinal calcium absorption. The right diagram illustrates a complex endocrine pathway for insulin secretion. A rise in blood glucose levels activates a sensory receptor, which signals the integrating center in the pancreas. The pancreas releases insulin, which targets tissues to increase glucose uptake and restore normal blood glucose concentrations. The posterior pituitary stores and releases two neurohormones. It is neural tissue and stores hormones produced in the hypothalamus. When the hypothalamus is stimulated, the posterior pituitary secretes vasopressin, also known as antidiuretic hormone, and oxytocin. The anterior pituitary secretes six hormones. It has an epithelial origin, making it a true endocrine gland. It releases trophic hormones that stimulate the secretion of other hormones. The anterior pituitary hormones include prolactin, thyrotropin, adrenocorticotropin, growth hormone, follicle-stimulating hormone, and luteinizing hormone. These hormones are regulated by hypothalamic trophic hormones. The pituitary is made up of two glands with different embryological origins that fuse during development. It sits in a protected pocket of bone and is connected to the brain by a thin stalk called the infundibulum. The posterior pituitary is an extension of neural tissue. The anterior pituitary is a true endocrine gland with an epithelial origin. The anterior pituitary is a true endocrine gland that secretes six classic hormones. Neurohormones from the hypothalamus control the release of the anterior pituitary hormones. The hypothalamic hormones reach the anterior pituitary through a specialized region of the circulation called a portal system. Neurons synthesize trophic neurohormones that are released into capillaries of the portal system. Portal veins carry the trophic neurohormones directly to the anterior pituitary, where they act on the endocrine cells. Endocrine cells release their peptide hormones into the second set of capillaries for distribution to the rest of the body. Prolactin targets the mammary glands. Thyroid-stimulating hormone targets the thyroid gland. Adrenocorticotropic hormone targets the adrenal cortex. Growth hormone targets the musculoskeletal system. Gonadotropins, including luteinizing hormone and follicle-stimulating hormone, target the gonads, specifically ovaries and testes. The posterior pituitary is an extension of the brain that secretes neurohormones made in the hypothalamus. Neurohormones are synthesized and packaged in the cell body of neurons located in the hypothalamus. These neurohormone-containing vesicles are transported down the axon to the posterior pituitary. Once they reach the posterior pituitary, the vesicles are stored until the neuron is stimulated. Upon stimulation, the neurohormones are released into the blood for distribution to target organs. The two primary neurohormones secreted by the posterior pituitary are oxytocin and vasopressin. Oxytocin targets the mammary glands and uterus, playing a vital role in milk ejection during breastfeeding and uterine contractions during labor. Vasopressin, also known as antidiuretic hormone, targets the kidneys to regulate water balance and blood pressure. The portal system consists of two sets of capillaries connected in series by a vein, facilitating direct hormone transport between the hypothalamus and anterior pituitary. In the hypothalamic hypophyseal portal system, hypothalamic neurons produce trophic hormones, which are released into the first capillary bed in the portal system and transported to the anterior pituitary. Anterior pituitary endocrine cells respond by producing their own trophic hormones, which are released into the second capillary bed and directed to target tissues. This system ensures that a small amount of concentrated hormone reaches its target efficiently. This diagram illustrates the hypothalamic anterior pituitary pathway, showing how the hypothalamus regulates the anterior pituitary gland through releasing and inhibiting hormones, which then influence various target endocrine glands and tissues in the body. The hypothalamus produces several hormones that control anterior pituitary function. Dopamine inhibits prolactin secretion. Thyrotropin-releasing hormone stimulates the release of thyroid-stimulating hormone and prolactin. Corticotropin-releasing hormone triggers the secretion of adrenocorticotropic hormone. Growth hormone-releasing hormone stimulates growth hormone release, while somatostatin inhibits growth hormone and thyroid-stimulating hormone. Gonadotropin-releasing hormone prompts the release of follicle-stimulating hormone and luteinizing hormone. Upon stimulation by these hypothalamic hormones, the anterior pituitary secretes hormones that act on various endocrine glands. Prolactin targets the breast, promoting milk production. Thyroid-stimulating hormone stimulates the thyroid gland to release triiodothyronine and thyroxine, which regulate metabolism. Adrenocorticotropic hormone targets the adrenal cortex to release cortisol, helping manage stress and metabolism. Growth hormone promotes growth and metabolism through the liver's production of insulin-like growth factors. Follicle-stimulating hormone and luteinizing hormone act on the gonads, stimulating the production of sex hormones, including estrogen, progesterone, and testosterone, and the development of eggs and sperm. The target organs and their responses include the breast producing milk in response to prolactin, the thyroid gland increasing metabolism through thyroid hormone release, the adrenal cortex managing stress via cortisol production, the liver generating insulin-like growth factor to promote growth, and the gonads producing sex hormones essential for reproductive functions. This interconnected system coordinates growth, metabolism, stress response, and reproductive processes through the integration of the hypothalamus, anterior pituitary, and target endocrine organs. Prolactin is a hormone primarily responsible for stimulating milk production in the female breast after childbirth. It plays a crucial role in maternal behavior and reproductive functions. Prolactin secretion is regulated by the hypothalamus through the release of dopamine, which serves as a prolactin-inhibiting hormone, ensuring that prolactin levels remain controlled unless stimulated by factors like breastfeeding. Growth hormone, also known as somatotropin, is vital for regulating overall metabolism, stimulating the growth of bones, skeletal muscles, and soft tissues, and supporting protein synthesis. It promotes the production of insulin-like growth factor 1 in the liver, which mediates many of its growth-promoting effects. Growth hormone secretion is regulated by the hypothalamus through the release of growth hormone-releasing hormone, which stimulates its secretion, and somatostatin, which inhibits its release to maintain balanced hormone levels. This diagram illustrates the growth hormone pathway and shows how the hypothalamus, anterior pituitary gland, liver, and target tissues work together to regulate growth and metabolism. The hypothalamus releases growth hormone-releasing hormone, which stimulates the anterior pituitary gland to secrete growth hormone. Growth hormone acts on many tissues in two primary ways. First, it directly stimulates various tissues to promote growth and metabolic functions. Second, growth hormone stimulates the liver to produce insulin-like growth factors. These insulin-like growth factors enter the bloodstream and target bone and soft tissues, where they promote cell division, tissue growth, and bone elongation. This pathway ensures coordinated regulation of growth, development, and metabolism throughout the body. The anterior pituitary gland produces two hormones called gonadotropins, follicle-stimulating hormone and luteinizing hormone. These hormones regulate reproductive functions and the production of sex hormones, such as estrogens and testosterone, in the ovaries and testes. The thyroid-stimulating hormone, also known as thyrotropin, controls the synthesis and secretion of hormones from the thyroid gland, including triiodothyronine and thyroxine. The adrenocorticotropic hormone stimulates the adrenal cortex to produce hormones, primarily cortisol, which is a steroid hormone involved in stress response and metabolism. The concentration of a hormone in the blood serves as the signal in a negative feedback loop. In long-loop negative feedback, a hormone produced by a peripheral endocrine gland inhibits the release of hormones from both the anterior pituitary and the hypothalamus. This mechanism is the most common form of feedback in the endocrine system, helping to maintain hormonal balance and prevent overproduction. Negative feedback in endocrine pathways maintains hormone balance in the body. When hormone levels rise, they signal the hypothalamus and anterior pituitary gland to reduce hormone production. In the cortisol pathway, the hypothalamus releases corticotropin-releasing hormone, which stimulates the anterior pituitary gland to secrete adrenocorticotropic hormone. This hormone triggers the adrenal cortex to produce cortisol. As cortisol levels increase in the blood, they inhibit the release of both corticotropin-releasing hormone from the hypothalamus and adrenocorticotropic hormone from the anterior pituitary gland. This feedback prevents excessive hormone production, protecting the body from harmful effects like high blood sugar or immune suppression. Similar feedback loops regulate other hormones, including those controlling thyroid function, growth, and reproduction. Hormones interact in three main ways to regulate body functions. First, in synergism, the combined effect of two or more hormones is greater than the sum of their individual effects. This enhances the overall response in the body. Second, permissiveness occurs when one hormone allows another to exert its full effect. For example, reproductive hormones control the maturation of sexual organs, but this process requires thyroid hormone. Without it, sexual maturation is delayed. Finally, antagonism happens when one hormone opposes the action of another. A common example is insulin, which lowers blood glucose levels, while glucagon raises them. These interactions ensure balance in growth, metabolism, and other essential processes. Synergism occurs when the combined effect of hormones is greater than the sum of their individual effects. On the graph, the y-axis represents blood glucose levels in milligrams per deciliter, while the x-axis shows time in hours. Individually, glucagon, epinephrine, and cortisol each cause a slight increase in blood glucose levels. However, when glucagon and epinephrine are combined, the increase is significantly higher. Most notably, when glucagon, epinephrine, and cortisol are combined, blood glucose levels rise dramatically, surpassing the effects of any hormone alone or even two combined. This demonstrates how synergistic interactions between hormones can amplify physiological responses, ensuring the body can meet increased energy demands during stress or fasting. Endocrine pathologies arise when there are disruptions in hormone secretion, leading to two primary conditions: hypersecretion and hyposecretion. Hypersecretion refers to an excessive release of hormones. This exaggerates the hormone's effects, often caused by tumors or medical treatments. Over time, negative feedback mechanisms can lead to the shrinking or atrophy of the hormone-producing gland. On the other hand, hyposecretion occurs when hormone levels are deficient. This may result from reduced synthesis or gland atrophy. Without proper hormone levels, the body lacks negative feedback, causing the brain to overproduce stimulating hormones. Both hypersecretion and hyposecretion can significantly disrupt bodily functions, highlighting the importance of hormonal balance in maintaining health. This slide illustrates how exogenous hormones, those administered from outside the body, can mimic the body's natural negative feedback mechanisms. When the body receives exogenous cortisol, it sends a signal that cortisol levels are sufficient, prompting the hypothalamus to reduce the release of corticotropin-releasing hormone. This, in turn, causes the anterior pituitary to decrease the secretion of adrenocorticotropic hormone. As a result, the adrenal cortex lowers its production of cortisol, while the exogenous cortisol still reaches target tissues and triggers the intended response. Prolonged use can suppress the body's ability to produce cortisol naturally. Understanding this process is crucial when managing therapies involving external hormone administration. This slide explains how receptor or second messenger issues can lead to abnormal tissue responsiveness. When hormone levels remain abnormally high, the body initiates downregulation by decreasing the number of hormone receptors. This is the target cell's way of reducing its sensitivity to excessive hormone stimulation. For example, in hyperinsulinemia, chronically high insulin levels cause a reduction in insulin receptors, which diminishes the cell's responsiveness to insulin. Conversely, upregulation occurs when hormone levels are low. In this case, cells increase the number of receptors to enhance sensitivity and maximize the hormone's effect. Understanding these mechanisms is crucial, as they play a vital role in maintaining hormonal balance and proper tissue function. This slide focuses on how the diagnosis of endocrine pathologies depends on where the dysfunction occurs within the hormonal reflex pathway. A primary pathology occurs when the problem is in the final endocrine gland that produces the hormone. For example, if the adrenal gland fails to produce cortisol, the pathology is primary. A secondary pathology originates in the pituitary gland. If the pituitary fails to release adrenocorticotropic hormone, it affects downstream hormone production. A tertiary pathology is due to dysfunction in the hypothalamus, which disrupts the entire hormone cascade. Identifying the level of dysfunction is essential for accurate diagnosis and targeted treatment. This figure explains hypercortisolism, a condition characterized by excessive cortisol levels, by illustrating 3 types of hypersecretion based on the source of dysfunction. In primary hypersecretion, the adrenal cortex itself is the problem. It produces excessive cortisol, which leads to low corticotropin-releasing hormone and adrenocorticotropic hormone levels due to negative feedback inhibition. In secondary hypersecretion, the issue lies in the anterior pituitary gland. Overproduction of adrenocorticotropic hormone stimulates the adrenal cortex to release excess cortisol. Here, corticotropin-releasing hormone is low, but both adrenocorticotropic hormone and cortisol are elevated. In tertiary hypersecretion, which is rare, the problem originates in the hypothalamus. An overactive hypothalamus produces high levels of corticotropin-releasing hormone, which raises adrenocorticotropic hormone and cortisol levels. Negative feedback fails, resulting in sustained hormone elevation. Recognizing where the dysfunction occurs is critical for diagnosing and managing hypercortisolism. The pineal gland, a pea-sized structure located deep within the brain, plays a key role in regulating the sleep-wake cycle. Historically, it was referred to as the seat of the soul, believed to control the flow of vital spirits. By the mid-20th century, Its purpose remained unclear until researchers discovered its role in producing melatonin. Melatonin is a hormone derived from the amino acid tryptophan. It is secreted in response to darkness and helps regulate circadian rhythms, promoting restful sleep. Although small, the pineal gland is crucial for maintaining the body's internal clock and ensuring healthy sleep patterns. This graph illustrates the secretion pattern of melatonin, often referred to as the darkness hormone. Melatonin levels remain low during daylight hours, but begin to rise in the evening, peaking between midnight and early morning. This increase promotes drowsiness and helps regulate the sleep-wake cycle. After reaching its peak around 2 to 4 a.m., melatonin levels gradually decrease as morning approaches, encouraging wakefulness during daylight hours. Melatonin is vital for aligning the body's biological clock with environmental light-dark cycles, ensuring optimal sleep quality and overall circadian rhythm regulation. In this chapter, we explored the essential aspects of the endocrine system and hormone function. We began with an overview of hormones, discussing their role in maintaining homeostasis and regulating various body processes. Next, we examined the classification of hormones, focusing on their chemical structures and mechanisms of action. We then explored how hormone release is controlled, emphasizing feedback loops and the hypothalamic pituitary axis. Hormone interactions, including synergism, permissiveness, and antagonism, demonstrated how hormones can work together or oppose each other. Lastly, we reviewed endocrine pathologies, highlighting conditions of hypersecretion and hyposecretion and their physiological consequences. Understanding these key concepts provides a foundation for appreciating how hormones maintain balance and influence health.